Thank you. It's my pleasure to speak to you today in this beautiful location. I'm really honored to be invited, and it's been great to meet all these people from all over the world. So today I'm going to talk to you about... There you go. All right, so I'm going to talk to you today about measuring synaptic connectivity using array tomography. So before I get started, I just want to make a comment that the Allen Institute, you've heard a lot from my colleagues, Vilas and Terry and Trigva, and you hear later today from Saskia about data resources that are available right now on the website. But of course, before you get to that point where you have a streamlined process has standard operating procedures and QC and fancy graphics on the website, of course, there's a lot of work that goes before that to build up these pipelines. I presently work on a project that's still in development, so I'm not going to tell you today about data that's available, but rather give you a preview about what might be available in a few years if the development of this project goes well. All right, so my talk is organized in four basic parts. First, I'm going to describe to you the problem of measuring cell type specific connectivity with an emphasis on how difficult and large a problem this is, and then describe a conceptual strategy to address the large scale of this problem that we call shotgun connectomics. Then in the next section, I'm going to describe the particular kind of technology that we're developing to try to address this conceptual problem, namely array tomography. I'll tell you how it works and some of the things that you can do. And then in the next section, I'll talk about some specific technical problems that have to be overcome in developing this system that we're working on at the Allen. And then finally, I thought I'd take a kind of review through the literature of some examples of papers where people have used array tomography to measure synapses more generally in the brain so that you could have an idea about how array tomography might be useful to you in your research. Okay, so what is the cell type specific connectivity problem? Well, quite simply, it's just we'd like to have an understanding about how are all the different types of cells in the brain connected to one another. And the final output of such an understanding could be summarized in matrices such as these. This is an example matrix from a recent publication from the Tilius lab, or actually a communication about a publication from the Tilius lab, which summarizes electrophysiological measurements of synaptic connectivity between different types of neurons. And so here, every row in this matrix represents a different type of postsynaptic neuron, and every column a different type of presynaptic neuron, and the size of the boxes represents the probability that these types of these two neurons are connected. Here at the Allen, we like to refer to these sorts of matrices as so-called W alpha beta matrices, where alpha and beta represent indices representing the cell type, kind of reflecting the kind of modeling community's use of Wij to represent how individual neurons are connected, but in this case reflecting how types of neurons are connected. Okay, so in that matrix, they had 13 different types of cells that were described based upon their differences in their electrophysiological and morphological types. Now, you heard yesterday from Vilas and Trigva about how single-cell transcriptomics is really changing our understanding about the diversity of cell types in the brain, and I think we have to face the fact that the kinds of matrices that we'd like to gather are significantly larger than the 13 by 13. And unfortunately, the problem grows at least quadratically with the size of the matrix. And in this slide, I'm going to kind of walk you through a calculation about exactly how large a problem this really is. There are lots of unknowns, and so I'm kind of using a bracketed approach to kind of describe something on the lower end of the problem and something on the kind of higher end of the problem. So if you had, say, 40 different cell types, this matrix would have 1,600 different entries. And if you wanted maybe just a coarse understanding of how probable it is that any of these different cell types are connected, say 10% precision, you'd need 28 measurements per entry. A measurement in this case represents a sample of 2 cells from a particular pair of cell types and just asking whether those cells are connected. Not even a measurement of a particular connection, but just asking whether they're connected at all. So that adds up to 45,000 entries across that cell type. On the other hand, if you thought there were many more cell types, maybe 80, maybe there's even more than that, if it were 80, it would be 6,400 entries. Maybe you want higher precision, 0.5% precision. That means you need about 600 measurements per entry. That adds up to 3.8 million measurements you'd have to do. Okay, unfortunately, the problem is actually much worse than that because in reality, most techniques that can measure conductivity don't have the ability to uniformly spread those measurements specifically across different cell types. In particular, there are very rare cell types. If a cell type, say there is a 1% rare cell type, most techniques are going to rely upon randomly stumbling upon one of those rare cell types and then post hoc identifying it. So if you only get the rarest cell type 1% of the time, then the probability that you sample from a rare cell type to a rare cell type is actually the square of that, so you only get that 0.01% of the time. And so if you only made those 45,000 measurements, you'd effectively actually only get 5 measurements from the rarest to rarest cell type. So that means in order to get back up to even the 28 measurements for the coarse measurement, you need to get back 284,000 measurements. Of course, this gets even worse if you think that it's even rarer. Say 0.1% means it's 0.001% of the time you get these rare to rare connections, which means you'd only get 4 from your 3.8 million for these rare to rare connections. So if you want to get back up to 600, you need almost a 500 million measurements in order to be able to fill out this matrix. So there's lots of unknowns here. There's lots of free parameters, but I think I've convinced you that the problem is either large or very, very large. Okay, so if you match up that kind of picture of how big a problem this is with the techniques that are out there for measuring conductivity, you can see there's a bit of a mismatch. You can place measurements of different techniques for conductivity along an axis, like I've done here, that kind of represents how dense a sampling you're doing in any particular sample. So multipatch electrophysiology represents one extreme of the spectrum, where in any particular experiment you're measuring maybe between 4 and 12 neurons per sample, and you can do a few samples per day per experimenter. So that's 24 to 264 measurements of conductivity per sample. So if you scale this up, this is something that can handle maybe thousands of measurements per year, depending upon how many resources you want to put at the problem. On the far end of the other spectrum would be dense cubic millimeter scale connectomics. In such an experiment, you'd take a cubic millimeter, it would have 100,000 neurons on it. If you could trace all of the neurons within that volume, you'd get 20 billion measurements of connectivity. Again, this is asking 20 million answers to the question, are these two neurons connected or not? And it's not clear exactly how long this will take, particularly because the tracing problem is not yet fully solved. But I think it's safe to say it's going to take a long time, maybe 5 to 20 years. So this represents a sort of low latency but very large measurement kind of approach. So the idea behind shotgun connectomics is that maybe there's an intermediate scale which could be more efficient in the long run for measuring connectivity. So if we had a technique that could assay connectivity of 100 or 2,500 neurons per sample, and you could do this relatively quickly, say a sample every 2 weeks, then you'd get between 20,000 and 12.5 million measurements of connectivity per sample. And if you scale that up to several, to the order of years, you can imagine getting hundreds of millions of measurements of connectivity. So how would you go about doing that? So that's what I'm going to outline for you next. So this is a kind of conceptual cartoon version of how we imagine this experiment would work. So you'd start with a sample of brain. You would sparsely label a subset of neurons, say between 100 and 2,500 neurons in that sample, with something that fills the entire neuron, very similar to what you heard in the previous speaker, and then image all the entire volume that contains those neurons, or at least something about their local connections, and then computationally trace 100% of the axons and dendrites found within that sample, and computationally segment out the individual neurons. And then the idea would be that you would use the full axonal and dendritic morphology of those neurons into categorize them into different types of neurons, as well as maybe other kinds of information that you could layer on in your imaging. And I'll get into what that might be later. And then finally, of course, you need to actually identify the sites of synaptic contact between these individual neurons so that you could get an anatomical assay of connectivity between all of the neurons in your sample. So this is the kind of conceptual idea about how we think shotgun connectomics would work. So in this section, I've kind of outlined for you what the problem of cell type specific connectivity is, and emphasized how large a problem it is, and given you a brief description about how shotgun connectomics as a concept could be used to try to address the scale of this problem. All right, so in the next section, I'm going to describe to you how the particular technology that we're trying to use and develop at the Allen Institute in order to be able to realize this conceptual idea. And that technology is called array tomography. And so obviously, if we want to be able to image individual neurons and 100% of the axons and dendrites of those individual neurons within a sample, you need a very high resolution imaging technique that's capable of also imaging individual synapses in the brain. And this is a very difficult optical microscopy problem. And to understand why, you have to speak a little bit about the basic physics about how microscopy images are formed. And of course, when you have an image volume, you have individual pixels, and you think of those pixels as corresponding to a particular location in the sample. But of course, that's not actually true. In any optical microscopy system, individual pixels actually represent sampling from a sort of weighted average of locations within your sample. And those weights, physicists would describe those weights as a point spread function of your optical microscope. This is an image of an empirically measured point spread function from an oil 1.4 Na numerical objective microscope. And that's practically about the highest resolution optical microscope that you can build. And as you can see from this image here, that it's over a micron tall and a few hundred nanometers wide. So the principle, this is why most optical microscopy techniques have a sort of elongated blur in Z, because this point spread function is typically longer in the optical axis than in the lateral axis. So the principle behind array tomography, furthermore, synapses in the brain can often be located closer to one another than a micron. And so it's difficult to use optical microscopy techniques that suffer from this point spread function to measure individual synapses. So array tomography starts, in fact, with a very similar principle to the knife edge, or to the sectioning imaging technique that we heard about from the previous speaker, which is that it is possible to physically section materials thinner than the point spread function of light, particularly in Z. So you can cut sections less than 100 nanometers. This has been known for many decades, as electron microscopy is sort of based upon using these sorts of techniques. And so here I've just kind of drawn a 100 nanometer scale bar to re-emphasize the point that you can do this. So array tomography starts with similar to the previous speaker, embedding your brain sample in a hard plastic resin so that you can collect ribbons of ultra-thin serial sections and collect them onto an optical cover slip. With the sections on this optical cover slip, you can then perform standard secondary immunofluorescence staining to highlight different molecular elements within this sample. Of course, we're particularly interested in synapses, so we typically use synaptic markers. And then you mount the sample on an automated fluorescence microscope and image each of the sections, and then take that imagery from each of the sections and reassemble it in the computer into a coherent volume. Furthermore, because the sections are embedded in this hard plastic resin, you can apply solutions to these things that you wouldn't normally think would be possible to normal brain tissue, such as very high pH solutions that can cause the antibodies to change their conformation and release from the sample. This then allows you to come back and restain the same sections with multiple rounds of different colors in order to build up a highly molecularly multiplexed picture of the molecular distribution within your sample. You can take all that data and reassemble it in the computer and form a volumetric image. So this is one example volumetric image that I'm showing different parts of, that I'm highlighting that highlights different parts of the neural circuit. So in green, you're seeing myelin basic proteins, so you can see all of the individual myelinated fibers within this sample. In red, you're seeing GABA, and so each of those little red balls represent a different GABAergic bouton. In cyan, you have neurofilament, which is showing individual specific fibers that use neurofilament. And again, you can trace those individual fibers. And actually, I think kind of quite interestingly, in cyan, you have DAPI. This very high resolution image of DAPI, you can see the ultrastructural, the heterochromatin of individual neurons. And you can even note in this small volume the kind of heterogeneity of that heterochromatin from neuron to neuron. This is just another movie that kind of zooms in and out and pans through another volume. Now here red represents synapsin, so each of those little red dots represents an individual presynaptic terminal. This is done in a mouse, YFP line H mouse, so you can see a subset of the neurons, their dendrites and axons, lit up in green. And then blue here is now tubulin, and so all of the large tubulin microtubule structures within dendrites can be seen in this image. So it kind of zooms in and out and pans back and forth to give you a sense of the volumetric nature of this data. All right, so then finally, you can also, after you're done doing your immunofluorescence imaging, you can post-stain these very same sections with heavy metals in order to take this cover slip and put it in a scanning electron microscope and take pictures of the very same sections. And if you do your tissue preparation accurately, you can get from that same movie that I showed you, myelin, this is a section from that very same block. And if you zoom in here, you can see all of the basic structures that you'd like to see in an electron micrograph. You can see the vesicles in this presynaptic. Does this work? No. Okay. You can see here the presynaptic vesicles. You can see the synaptic cleft. You can see a spine and a spine apparatus. All right. So because you can image these same sections in these two different modalities, you can actually integrate this data in a remarkable way. So here I've got a picture of a ribbon, an overview image in the SEM, and an image in DAPI. There we go. So I can now actually overlay these two images and begin to zoom in on an individual section. So as we zoom in here, you can start to appreciate how precisely you can actually register these two imaging modalities. So this is the peel surface here. You can see the individual DAPI nuclei line up right on top of them. Within this box here, I've actually got multi-channel immunofluorescence imagery at higher resolution, and then within here, there's higher resolution EM. So as we zoom in, you can see green now is the synaptophysin, which is a part of the presynaptic apparatus. Red is PST95, which is right on top of this cleft here. And GABA is in blue, and so you can see these GABAergic processes spread out. So I'm just going to kind of show another set of images that illustrate the same point. Here's now an image of a section in which there is high resolution immunofluorescence throughout the entire section. You can see some individual GABAergic cells spread throughout this thing. And then within here, there's higher resolution EM. So as we zoom in, you can see DAPI really precisely lines up over them. And if we zoom in even further, you can, now I'm showing you just the electron micrograph, so you can appreciate synapses. Here's a synapse, presynaptic zone, postsynaptic zone. Here's a myelinated axon. And if I layer on the immunofluorescence, you can see how precisely the immunofluorescent signal, say PST95, lies up right on top of that cleft. All the presynaptic zones are filled with synapsin, and myelin is tracing right around the myelinated processes within this section. So of course I'm showing you there 2D, but of course you can still be doing this in 3D. So here's an example just of 3 sections of YFP that allows you to see that this spine right here is obviously a YFP positive spine without having to produce any kind of DAB reaction within your electron micrograph. Okay, so to summarize the advantages of array tomography, it's a very high-resolution imaging technique, as I discussed. It effectively produces focal volumes that are 100x smaller than a confocal microscope. It has the ability to multiplex many different molecular signals within an individual experiment. And then this uniformity is an important point. You have, by physically sectioning these ultra-thin sections, you give kind of uniform exposure of reagents as well as excitation light and emission light from your microscopy. Although I showed you that point spread function that was a 1 micron tall and a few hundred nanometers wide, and that's a sort of close to the theoretical limits. Most practical microscopy situations, such as imaging a confocal microscope into a thick section, have point spread functions which are much, much worse than the theoretical limits. And that's because imaging into brain tissue is scattering, it has non-uniform index of refraction, and all of these things cause the point spread function to be degraded. When you image something directly on the opposite side of an optical cover slip, you actually get very close to the diffraction limit. And you have very good access of your illumination, and you get to collect all of the photons without a tremendous amount of scattering. And then furthermore, you're exposing each of the sections to the same concentration of antibody reagents, and so there's sort of better internally consistent immunostaining throughout the volume compared to kind of volumetric labeling methods. Finally, it's very fast because it's a wide field fluorescence microscopy technique. You collect whole frames at a time. We typically use SCMOS cameras that will capture a 200 micron square area in a single snap in a few hundred milliseconds. And so you can collect a tremendous amount of data. It has relatively high signal to noise, again, because you're able to use such a high numerical aperture objective. You can collect a larger fraction of the fluorescence that's produced by your sample. And as I've shown you, it is sort of multimodal or correlative or conjugate in that you can combine the immunofluorescence and electron microscopy quite precisely. Of course, has its disadvantages. First and foremost, it's a fixed imaging technique. It's not something that lets you look at the dynamics of the brain. Importantly, it relies upon using specialized antibodies. When you take your tissue and you embed it in these hard plastic resins, it goes through a series of tissue processing steps which can change the conformation of proteins. When you're using antibodies, antibodies recognize specific antigens on those proteins, and if those proteins are changed in some way, it can make the antibodies ineffective at recognizing those sites. And so we've had to do a lot of work to find antibodies, antibody-protein combinations that work in this specific context. It also requires some specialized equipment and processes like ultramicrotomes and to be able to collect these ultra-thin sections. And finally, it's computationally intensive and it produces large amounts of data. Similar, again, to the previous speaker, there's a large number of image processing steps, which I'll get into in detail a little bit later. All right, so in this section, I've kind of summarized for you what array tomography is and how it works at a basic level. So in the next section, I'm going to describe to you the kind of technical problems that are associated with doing array tomography, particularly if you're trying to do array tomography at a very large scale that's necessary to do shotgun connectomics. So the first problem you're faced when you're trying to do imaging is you put one of these ribbons on your microscope and you want to take a picture of the same corresponding region in each of the sections. But you don't know exactly where are those sections laid out. So this is a kind of movie that illustrates software that we developed at the Alum for automating that process. So you start by identifying a location within a section that you'd like to image and roughly where it is in the next section, and it uses basic computer vision techniques to be able to refine the position of that second section. And because the sections are laid out in a relatively reproducible manner, it's able to use the vector between two adjacent sections to estimate where the next section is and then use computer vision to refine that. And you can see here, this video, it's able to basically automatically map all of these sections in this ribbon. This is sped up about 4 times real time, but nonetheless, it's able to do it quite rapidly. And then once you have a map, then you want to plan a mosaic of locations which image, in our case, is the entirety of the section. And so this software helps you do that. Each of these little cyan boxes represents an individual XY location that your microscope is going to go before it snaps a picture. And you can see it's actually laid out this mosaic in a way that respects the slight change in orientation of the sections as it goes through the different layers of the ribbon. And then it will actually execute imaging. So here's a short clip that shows you our microscope in action at the Allen. Each flash of light is a single frame, and this is a real-time movie. So each flash of light represents a single frame of a 4-channel immunofluorescent image taken at each XY location. You can see the microscope runs at about 4 hertz. Each image is 8 megabytes, so it takes data between 30 and 40 megabytes a second. And now we have 3 such systems at the Allen, so this will really produce a fire hose of imaging data. Of course, our problem here is not imaging just a single ribbon, but actually collecting hundreds of ribbons in order to form these large-scale images that we want to do. And effectively, one of the limitations on throughput is that if you're doing one ribbon at a time, you need a human there to put the ribbons on and to do the staining. And that effectively means your microscope isn't running 24 hours a day, and so you're leaving a factor of 4 or 5 on the table of throughput. So we're trying to develop a system to allow the microscope to run 24 hours a day by integrating a pipetting robot onto the robot to integrate imaging and staining at the same time. And so we're doing that by building these sorts of flow cells here. Here's an example of one of our cover slips that's been carbon coated to provide adhesion of the ribbon and connectivity for scanning electron microscopy. We mount it together with this flow cell, which I'll describe in more detail in a minute. And this flow cell basically allows the pipetting robot to apply reagents to the ribbon without the use of any, in a sort of passively pumped manner. So there's this, you make use of this principle. It was published in this paper. Basically, you have a small hole and a large hole, and the difference in surface tension between the small hole and the large hole allows passive pumping of reagents through this flow cell. And so this way, you can see this is actually a pipetting robot that is able to put reagents in this hole and take them out of this hole. So this is how we're hoping to build a kind of on-microscope immunostaining system. And so in the end, we hope to set up something like this, where we mount up 8 ribbons at a time and have the microscope imaging one ribbon while the rest are getting immunostained, and have them sort of ready for imaging in a just-in-time manner that will allow continual cycles of stripping and staining and imaging to happen in a process that hopefully will take up most of a 24-hour cycle. Okay, so I've described the basic problem of large-scale imaging. Of course, you have image processing problems to solve as well. So I'll walk through the 3 imaging processing problems that we have. The first is sort of familiar, I think. Here's a kind of 3x3 mosaic of individual frames. You can see you have some overlapping regions. I'll make some of them red and some of them green. And it looks pretty good from this point, but if you zoom in here on this one nuclei, you can see that there's a slight shift here in this nuclei. It's not perfectly registered between these different frames, and this is because there's some imprecision in the stage of your microscope, so you need to computationally shift these images to form a coherent montage. The second problem that you have is that if you'd like to do multi-round imaging in order to combine, say, here's an example 3-round imaging plan for imaging some typical synaptic markers. You'd like to register all this data into the same space. Our strategy for doing this is to repeat one of the stains across the sections, and we typically use DAPI because it's so easy to apply and gives us such a nice reliable signal. But if you look at, say, 2 channels of DAPI, say the channel 1 and one from session 1, one from session 2, you're never going to be able to make the microscope go back to exactly the same XY location, particularly if you took the sample off the microscope and put it back on, and so you have the images don't line up perfectly. On the other hand, they were attached to this rigid optical cover slip, and so they do really align with a simple rigid transformation. And so you're able to use we can use computer vision techniques to identify corresponding features in these 2 images and then transform one into the other to make them line up quite perfectly. And now I've done that here. And you can see it basically looks exactly the same, although this is the merging of 2 separate images that were taken on 2 different imaging sessions. So those are the 2 sort of simpler problems. Of course, the more difficult problem is what I call the alignment problem. I end up getting into arguments with people about what to call each of these different stages. But here is a kind of movie in which each frame of the movie represents a different Z section, and this is just raw data that's been stitched but hasn't been aligned. And you can see the problem is that they are not perfectly lined up. You can again use those similar computer vision techniques to find corresponding features and calculate a linear alignment between these different sections, and that works pretty well. But if you look very closely, you can see here as I zoom in on some section of DAPI, you get again a little bit of jitter, and this is because the sections, as they're sectioned, and particularly as they are laying down onto this optical cover slip, can get stretched slightly in nonlinear ways. And so you need to calculate a nonlinear transformation across sections to bring all of this data into alignment. So this is a problem that has faced electron microscopy for years, and we use, in fact, the very same algorithms that were developed for doing this kind of alignment. In particular, we use this algorithm that was developed by Stefan Saalfeld at Genelia Farm. I'm not going to go into details about how it works precisely, but in short, it sets up the problem as a relaxation of problem of this very large network of springs, which connect points of correspondence between different sections. And then you relax this spring network in order to find the nonlinear transformation to bring the data into register. So here's an example of that same data set after this nonlinear elastic alignment has been applied to it. And now if I zoom in on an individual region, you can see that the data can be lined up quite precisely. So once you're finished doing all of those image processing steps, you end up with coherent three-dimensional data in many different sections. That's kind of difficult to look at, particularly if you have many different colors. It starts to look like Skittles if I show it colorized. So we like to look at individual synapses like this. So here I'm showing a typical glutamatergic and GABAergic synapse, and each column here represents a different Z section. And the different rows represent different molecular channels. Here up top, I've shown a zoom in of one of the sections in the electron micrograph. You can see this is the presynaptic area and the postsynaptic area. Similarly here, presynaptic, postsynaptic. And if I zoom in a little bit on this section, you can see I've highlighted in green the presynaptic zone and in red the postsynaptic zone, and then repeated that across the channels in order to give you some sort of spatial reference. You can see here that you're seeing the kind of molecular hallmarks of different types of synapses. So the excitatory synapse has got PSD95 in the synaptic cleft. It's got some NMDA receptor signal. It also has VGlut1 in the presynaptic zone, and it has synapsin in the presynaptic zone, and it has an absence of inhibitory markers. On the other hand, the inhibitory synapse here doesn't show any of the inhibitory markers, but it shows, again, synapsin in the presynaptic zone, as well as gefrin, a marker of the postsynaptic process of the postsynaptic specialization in inhibitory synapses, and GAD and GABA in the presynaptic zone of inhibitory synapses. So that kind of shows you anecdotally that you can use array tomography to look at the molecular composition of individual synapses, but you'd like to have a quantitative method to localize and count all of the synapses in your sample. So that's what I'm going to walk you through in these slides here. So here I'm showing you a single field of view of PSD95 immunofluorescence along with its corresponding field in the electron micrograph. And I'm going to show you this example all in 2D, but you have to keep in mind that really this is a three-dimensional problem, and I have three-dimensional data. So the green circles here are the result of a very simple threshold on the immunofluorescence data and finding the connected components that exist above a threshold, and I've repeated those circles over here for your reference. And here I've manually annotated each of the synaptic clefts that are visible in the EM and colored them with respect to whether they're GABAergic or non-GABAergic. Of course, most non-GABAergic synapses are glutamatergic. And as you can see, most of the PSD95 immunofluorescence lies on top of synaptic clefts, although there are some examples of non-synaptic PSD95 immunofluorescence. This can happen for a number of reasons. First, because there's some level of non-specific binding of antibodies, but second, because PSD95, like all proteins in the brain, must be produced somewhere and transported to and from synapses, and so you wouldn't expect that 100% of PSD95 exists at synapses. The challenge for an algorithm you'd like an algorithm which analyzes only the immunofluorescence data, because that data is much more easy to acquire and can be acquired at much larger scales much more quickly, and is able to interpret this and accurately predict where there are synapses. And so you can use a machine learning technique that uses this electron microscopy as the kind of ground truth to train up an algorithm that uses only the immunofluorescence as its data. So in this plot over here, I'm quantifying each row represents one of those PSD95 immunofluorescence spots, and each column represents a quantification of the immunofluorescence signal in a number of different channels. The channels here in red are channels which are useful for measuring the difference between PSD95 objects which are at synapses, this is lower half, versus not at synapses, which is this upper half. And I've also got columns which quantify the number of sections that the PSD95 immunofluorescence is in, as well as the total area. And as you can see, there's many channels which are predictive. And you can try to combine all all these channels of information to train a machine learning algorithm, such as a SVM classifier, to tell the difference between a PSD95 spot that's at a synapse versus one that's not. And you can do so quite effectively. You can get, this SVM was able to learn to correctly identify about 95% of the synapses while having a less than 10% false positive rate. Okay, so in this section I've kind of summarized for you some of the technical problems that have to be solved in doing array tomography, particularly on large scales, including that you have to automate your imaging and that this produces large amount of data quite quickly, that these raw images must be processed through a multi-stage image processing pipeline, and then you have to analyze these data in order to be able to find all the individual synapses. I didn't talk about the very significant problem of needing to trace these synapses, trace the individual neurons, but that is, as the previous speaker mentioned, a very important and challenging one as well. All right, so in the final section, I thought I would take a tour through the literature to kind of describe how some other groups have used array tomography to measure synapses in the brain more generally, not necessarily in the context of shotgun connectomics. So I thought I'd start here with a kind of classic example. People use this technique simply for counting synapses and measuring changes in the density of synapses following some manipulation. So in this particular paper that was published in Neuron, they looked at the effect of knocking out this cannabinoid receptor on the effect of formation of synapses during development, and found we're able to use array tomography to quantify that the knockout animal had a larger number of synapses in the thalamus in this particular experiment. So that's a kind of simple use case for just using array tomography to measure changes in synaptic density. Of course, it's an imaging technique, so you can use this to look at differences in the spatial distribution of synapses. So here's an example from a paper that was measuring how synapses are affected based upon the distance from plaques in an Alzheimer's model of mice. So here they were able to use a antibody to beta amyloid to visualize the plaque, and then measure the synaptic density as a function of distance from the plaque. And of course, within the plaque, there are very few synapses, but in fact, you can see that synapses are significantly affected up to a few tens of microns away from the plaque location. And then finally, I thought I'd spend a little bit of time emphasizing this paper that I think is quite impressive and interesting, from Eric Bloss and Nelson Spruceton at Genelia Farm that was published in Neuron just a few months ago. And in this paper, they used array tomography to quantify how different types of interneurons innervate different components of the dendritic arbor of excitatory neurons in the hippocampus. So their strategy was to make samples in which they had lit up the excitatory neurons in one color, and then lit up different subtypes of interneurons in another color using a transgenic strategy. And in this way, they were able to reconstruct the complete dendritic arbors of individual excitatory neurons, and then quantify as a function of different types of different locations of dendrites, say in different layers of the hippocampus, and different levels of primary versus terminal or intermediate dendrites, how these different subtypes of interneurons innervated in each of these different zones. So there's a lot of data here. I don't have time to go into it all in incredible detail, but suffice it to say there are significant differences between the different subtypes of interneurons and where they innervate these pyramidal cells. And then what I thought was particularly interesting is that they were able to now take this data and use it to inform computational models of these excitatory neurons. So they took these morphologies and then built detailed biophysical models of these neurons, and then placed synapses of different specific inhibitory subtypes at locations within these morphologies consistent with the data that they had measured, and then studied the effect of applying excitatory inputs at different locations while simultaneously simulating the effect of different subtypes of synapses from different interneuron subpopulations. Again, they have a lot of different findings that describe this in detail, and I'm not going to go into it. You should read the paper if you're interested. But suffice it to say, they were able to show that the the specific innervation of different subtypes at different regions in the dendrites have significant effects on how these dendrites process and perform dendritic integration, and that different subtypes have different functional roles, which are assisted by the localization of where those subtypes innervate on these excitatory cells. So in this section, I've kind of given you a brief outline of some different papers that exist in literature and how they are using array tomography to measure synapses in the brain, and how array tomography can be used to measure the spatial distribution of synapses across different biological structures, including layers, distance from plaques, and their subcellular localization. So in conclusion, as with all the speakers and everyone who works at the Allen, this is really a very large team project, and so there are many, many people to thank that I don't have time to thank everyone individually. We also have collaborators at Johns Hopkins University who help us with the bioinformatics of processing this data, and the University of North Carolina in tissue prep, and at Stanford still, Christina Makeva is the co-inventor of array tomography, and we still work quite closely with her. So with that, thank you for your attention, and I'd be glad to take any questions.